Have you ever seen a biological phenomenon, maybe something like a glowing insect or even just a muscle flexing, and it hits you that what you're watching it's fundamentally a physics problem?
It absolutely is.
I mean, how does a cell, which is essentially just a bag of molecules and water, manage the constant energy crisis that's required just to stay alive?
It is the ultimate logistical challenge for sure, and it's all governed by rules that are much, much older than life itself.
I was really struck by this image from our sources. It's this almost alien sight of green glowing larvae of a click beetle on a termite mound in Brazil.
Oh, bioluminescence, it's beautiful.
It's incredible. But that light isn't magic, right? It's just chemical energy being converted into light energy. Also, it can attract its dinner.
And that conversion is exactly what we're here to dive into. We're looking at what's called bioenergetics, the study of how energy flows through living systems. The whole collection of chemical reactions in an organism is its metabolism.
So our mission today is to unpack that, to get into this huge, organized flow of energy from the big universal laws that dictate it all the way down to the tiny molecular machines that actually do the work.
Exactly.
Okay, let's unpack this. If metabolism is the total sum of all these chemical reactions, how on earth does the cell keep them all straight? It sounds like a logistical nightmare.
It would be, but it's less of a nightmare and more of an extremely elaborate, highly coordinated roadmap. Metabolism is made of all these intersecting metabolic pathways.
Okay, a roadmap.
Yeah, or think of it like a production line. A specific starting molecule gets altered through a series of really well-defined steps, and this is the crucial part. Each of those steps is overseen by a different specialized enzyme.
And these pathways, they generally run in two different directions, right? You have the ones that break things down and the ones that build things up.
Precisely. First, you've got the downhill routes. Those are the catabolic pathways. They release stored energy by breaking down complex molecules.
Like cellular respiration.
Cellular respiration is the textbook example. You're taking glucose and breaking it down into simpler stuff. That's a major energy releasing process.
So the opposite must be the anabolic pathways, the uphill routes.
Right. Those are the biosynthetic paths. They consume energy to build complicated molecules, like, say, taking individual amino acids and stringing them together to synthesize a new protein.
So the energy you get from the catabolic breakdown is the currency you spend on the anabolic building projects.
That's the cycle. Life is this constant process of tearing down and building back up.
Okay, before we get to that currency, let's just do a quick review of the fuel itself, energy. We all know kinetic energy, that's the energy of motion.
And that includes things like thermal energy, which is just the random motion of atoms and molecules, heat.
But for biology, the really interesting one is potential energy. That's the stored energy, based on an object's location or its structure.
And in the context of a cell, we're almost always talking about chemical energy, which is just the potential energy stored inside chemical bonds.
The classic analogy here is the diver on a platform, right?
It's perfect. As the diver climbs the ladder, her kinetic energy of movement is converted into potential energy because of her height.
More height, more potential energy.
And when she dives, all that stored potential energy converts right back into kinetic energy. So in a cell, a molecule like glucose is the diver on the high platform. It's packed with potential chemical energy.
And the products, after it's broken down, are like the diver who has just hit the water. They're at a lower, much more stable energy state.
Exactly. And all of these transformations, every single one, has to obey two unbreakable cosmic laws, the laws of thermodynamics.
The first one is pretty straightforward. The first law is just the conservation of energy. You can't create or destroy it, only transform it.
Right, so a plant isn't making energy from the sun, it's just transforming light energy into the chemical energy stored in sugar. When a bear eats a fish, it's transforming the chemical energy from the fish into the kinetic energy of running.
But the second law, that one feels a lot more profound and, honestly, a little bit grim. Every single energy transfer increases the entropy, the disorder, the randomness of the entire universe.
Always. And the reason for that is that with every transformation, some of that useful energy is lost. It just escapes as heat, which is unusable for work. And that increase in random thermal motion directly contributes to the universe's total disorder.
So this means any process that's energetically favorable, what we call a spontaneous process, has to, by definition, increase the universe's entropy. But that seems like a huge contradiction for life.
How so?
Well, look at the incredible order in a living thing. We build these highly complex structures like a cell or that Venus flower basket sponge mentioned in the text. How can we create and maintain all that local order if the universe is always moving toward chaos?
Ah, that's the key. The organism is an open system. It isn't living in a sealed box. We pay for our incredible internal order by dramatically increasing the disorder of our surroundings.
We export our entropy.
We export entropy. We take in highly ordered things like food, and we release very simple, disorganized waste products, carbon dioxide, water, and crucially, waste heat. That heat dramatically increases the entropy of the environment.
So on a global scale, the second law is still satisfied. We're just borrowing order.
We are borrowing order from our surroundings to build ourselves up. That is the fundamental constraint of being alive. Now, if you want to predict which reactions are actually possible within that constraint, we have to turn to a concept called free energy.
Right, symbolized as G. And free energy is the portion of a system's energy that's actually available to do work, the useful part.
Yes, and what really matters to a cell is the change in free energy, which we write as delta G, or delta G. The full equation is delta G, delta H, T delta Se, but the take-home message, the most important rule for the cell, is this. Only processes with a negative delta G are spontaneous.
A negative delta G, so that's the dividing line. A reaction that can happen on its own versus one that needs a huge push of energy to happen at all.
Exactly. You can think of G as a measure of instability. A system with high G is unstable, like a complex molecule just waiting to break down. Spontaneous change always moves a system towards stability, which means toward a lower G.
And the most stable point of all is equilibrium. If a system reaches equilibrium, its G is as low as it can possibly be, and it can do zero work.
Yeah.
It's energetically dead.
Which gives us our two major types of reactions. If the delta G is negative, the reaction is exergonic. It means energy outward. Energy is released, it's spontaneous. Cellular respiration, for example, is strongly exergonic. It releases a lot of energy.
And if the delta G is positive, it's endergonic, or energy inward. It absorbs free energy from the surroundings, and it is absolutely non-spontaneous.
Right. Building glucose through photosynthesis is a classic energonic process. It needs a huge energy input from sunlight.
This all leads to maybe the most important point for a living cell, which is that metabolic pathways have to constantly avoid reaching equilibrium.
They must. If a cell's reactions ever hit equilibrium, all work would stop. That's what death is, thermodynamically speaking.
So how does the cell escape that fate? How does it dodge this thermodynamic death sentence?
Because it's an open system. Think of a hydroelectric dam. If you just seal it off, eventually the water level on both sides will equalize. That's equilibrium. The turbines stop. No more work. But a cell is like a dam with a river constantly flowing through it. It's always taking in new high-energy reactants like glucose, and it's constantly getting rid of low-energy waste products. This constant flow keeps the pathways far from equilibrium and allows the work of life to continue.
And to do all that work, it needs a universal energy currency, which brings us, of course, to ATP. How does the cell actually do this energy coupling? How does it use an energy-releasing reaction to power an energy-requiring one?
That is the specific job of ATP adenosine triphosphate. It's the molecular shuttle that powers almost all of the cell's work. We're talking chemical work, transport work, mechanical work, you name it.
So what's so special about its structure? What makes ATP such a perfect little battery?
It's all in the tail, the triphosphate tail. You have adenine, a ribose sugar, and then this chain of 3 phosphate groups.
Okay.
And here's the thing. All 3 of those phosphate groups are negatively charged. So they're all repelling each other, crammed together. It's like trying to hold the negative ends of 3 strong magnets together in your hand.
It's an unstable, compressed spring.
That's the perfect way to think about it. So when the bond to that last phosphate group is broken, when ATP becomes ADP plus a phosphate, the system moves to a much lower, more stable energy state, and it releases a significant amount of energy.
But how does that energy actually get transferred? It's not like the ATP just explodes next to another reaction, right?
No, that would just be wasted as heat. It's much more elegant. ATP usually drives reactions through a process called phosphorylation. It directly transfers that unstable phosphate group to another molecule.
So it creates a phosphorylated intermediate.
Exactly. And that intermediate is now energized, it's less stable, it's more reactive than the original molecule was. By slapping that phosphate group on, ATP has basically increased the free energy of the reactant, making the next step in the reaction energetically favorable.
So that's how a protein can change shape to pump something across a membrane. It gets phosphorylated.
That's how. And what's really mind-boggling is the pace of the ATP cycle. This isn't a single-use battery. It's constantly being
The
recharged.
numbers are just staggering.
They are. The catabolic pathways, like breaking down glucose, provide the energy to reattach that phosphate to ADP, regenerating the ATP. A single working muscle cell recycles its entire pool of ATP. We're talking 10 million molecules in less than a minute.
That is just a furious pace, and that's just to keep things going. So if breaking down glucose is so favorable, so exergonic, why doesn't a sugar cube just burst into flames when I drop it?
Right, that's the final piece of the puzzle. It's the activation energy, or ISAB.
The energy barrier.
The energy barrier. Even spontaneous reactions need an initial investment of energy to get over this hump. The activation energy is what's needed to contort the reactant molecules into a very unstable shape called the transition state, where their bonds are actually ready to break.
Like needing a spark to light gasoline. In the body, though, we can't just use heat, so we use enzymes.
Precisely. An enzyme is a biological catalyst, usually a protein, and its one and only job is to speed up a reaction by dramatically lowering that activation energy barrier.
And it's crucial to remember, they do not change the overall delta G. They don't make an impossible reaction possible. They just make a possible reaction happen on a time scale that's useful for life.
They make life possible, period. Without them, metabolism would be so slow it would essentially stop.
And their genius is their specificity. It all comes down to their unique 3D shape. The reactant, the substrate, fits perfectly into a specific groove on the enzyme called the active site.
And it's not a rigid lock and key. It's a dynamic process called induced fit. When the substrate binds, the enzyme actually changes its shape slightly to get a better grip. It's like a firm, clasping handshake.
So how does that handshake actually lower the energy barrier? What's it doing in there?
It's doing a few things at once. For one, it can act like a molecular matchmaker, just holding two substrates in the perfect orientation to react. It can also physically strain the bonds of the substrate, sort of bending them and making them easier to break.
Okay, so it's an active process.
Very active. It might also create a special microenvironment inside the active site, maybe a little acidic pocket that helps the reaction along. And sometimes it will even form a temporary covalent bond with the substrate itself.
It's an incredibly sophisticated machine.
It is. And its efficiency is very dependent on local conditions. You can speed up a reaction by adding more substrate, but only to a point. Once you hit saturation, when all the active sites are busy, the only way to go faster is to make more enzyme.
And of course, there are things like temperature and pH. Most of our enzymes work best at body temperature. A high fever is dangerous because it starts to break down or denature those precise protein shapes.
Right, but then you have amazing exceptions like the enzyme pepsin in your stomach, which thrives in a super acidic environment that would destroy most other proteins.
Enzymes also need helpers sometimes, right? Cofactors.
They do. These are non-protein helpers. They can be metal atoms like iron or zinc. If the cofactor is an organic molecule, we call it a coenzyme. A lot of vitamins function as coenzymes, which is a major reason why we need them in our diet.
And just as we can help them, we can also block them with inhibitors.
Yes, and there are two main types. You have competitive inhibitors, which are basically substrate lookalikes. They compete for the same parking spot, the active site.
You can overcome that just by flooding the system with more of the real substrate.
You can, but then you have the more dangerous ones, non-competitive inhibitors.
They don't bind to the active site.
Correct. They bind somewhere else on the enzyme, but in doing so, they change the enzyme's overall shape, and that change makes the active site less effective, or maybe not effective at all. Many poisons and nerve gases work this way. They're irreversible inhibitors.
Which brings us to the final point. With all these pathways and enzymes and inhibitors, how does the cell maintain order? It can't just be chemical chaos. It must regulate all this activity with incredible precision.
And that precision is achieved mainly through something called allosteric regulation. This is where a molecule binds to a regulatory site on the enzyme that is completely separate from the active site.
Okay.
Many of these enzymes oscillate between an active shape and an inactive one. So an allosteric activator can bind and lock the enzyme into its active form. An inhibitor does the opposite. It binds and stabilizes the inactive form.
That's such an elegant switch.
It is. A perfect example is how the cell manages its energy supply. When ATP levels are high, ATP itself acts as an allosteric inhibitor for some of the key enzymes in the catabolic pathways that produce ATP.
So it's a negative feedback loop. We have enough energy, slow down production.
Exactly. And conversely, if ATP levels fall, ADP levels rise. And ADP acts as an allosteric activator, signaling those same pathways to ramp up production and make more ATP. It's real-time supply and demand.
There's also cooperativity, right? Where one substrate binding makes it easier for others to bind.
Yes, that's another form of allosteric activation. One substrate binds, and it causes a shape change across the entire enzyme, making all the other active sites on it more receptive. It amplifies the response.
And maybe the most common control mechanism is feedback inhibition.
Absolutely. This is where the final end product of a long metabolic pathway loops all the way back to the beginning and inhibits one of the first enzymes in that pathway.
So once you've made enough of what you need, you shut down the entire assembly line right at the start. You don't waste any resources making intermediates you no longer need.
It's the ultimate in metabolic efficiency. And the cell boosts that efficiency even more by organizing things in space, either grouping enzymes that work together into complexes or sequestering them inside specific organelles, like how the enzymes for cellular respiration are packed inside the mitochondria.
So we've really mapped out the whole energetic framework. We started with the big universal laws of thermodynamics. We brought in ATP as that essential, unstable currency for doing work. And we ended with the enzymes, these incredibly precise catalysts that are regulated with such fine control.
That's the whole picture, from the universal to the molecular.
It is just astonishing to think that all of this, from a glowing beetle to our own muscles, has to constantly fight against that relentless second law of thermodynamics. That we have to increase the universe's total disorder just to maintain the incredible order inside a single cell.
And that fight is constant and incredibly expensive. We mentioned that one muscle cell recycling 10 million ATP molecules every single second. So this raises a final question for you to think about.
Sure.
Considering this massive, unrelenting energy cost that's required just to push back against universal entropy, what are the ultimate physical limits to how complex and how energy demanding a living system can possibly become?
A great question to ponder. Thank you for joining us on this deep dive into the source material.